Okay, are there any questions? All right, let me remind you, can you guys hear me? Let me put this on. Let me remind you that I sent out the unknown numbers. So you have your numbers and you have your first results. You should look at your results, look at the table, and then decide which test you want to run next and put it in your unknown notes page. Any questions about any of that? All right, I'm not hearing anything. Can somebody please confirm that you're hearing me? Okay, I'm not hearing anything. Let me see if there's any pictures. Let's close that down. Yeah, I don't have anyone with pictures, so I can't confirm that anyone is hearing me. Can anyone hear me?
We can hear you.
All right, thank you. Oops, shut that down. Okay, today is August 4th, and we should be finishing our lecture. I'm going to go up the syllabus now. Oops, didn't get there in time. So we should be finishing chapter 5 today and actually starting chapter 6. Any questions about any of that? I'm sharing my screen now. So hopefully we'll finish chapter 5 and start chapter 6. We do have a lab today. No, I take that back. Yes, we do. We do have a lab today. All right, any questions about anything? If not, let's begin. So we had talked about aerobic respiration and the electron transport chain last time. Is that right, or did we get further than this? Let me see what the next slide is. Seems to me we talked about this. Let me see if there's another 2 here. That's where I've got the only marker. Is that correct? Is that the slide we were on? Nobody's going to confirm this? All right. So in eukaryotes, we get 32 to 34. You know, I swear we did this. 32 to 34.
Yes, I think we've been over this.
We have been over it.
Yep.
I don't have a marker for where we stopped. Do you remember where we stopped? It's unlike me to not mark it, but
I think it was this slide, this one.
this one here,
Hmm.
let me look around. I'm pretty sure we covered this too. Did we talk about anaerobic respiration last time?
No, I don't believe so.
Okay. So this slide is showing you a summary of aerobic respiration and fermentation on the right. I'll blow this up. So in aerobic respiration, recall that we have 3 steps: glycolysis, which we generate 2 NADH, 2 pyruvic acid, and 2 ATP, which is followed by the preparatory step and the Krebs cycle, which generates 2 more NADH. And the Krebs cycle generates 6 NADH and 2 FADH2s. The NADH and the FADH2 carry electrons from glucose or the derivative of glucose to the electron transport chain. And then the electrons are pumped through a chain of proteins in a membrane, pumping hydrogen ion across the membrane, and then the hydrogen ion moves across at ATP synthase, making ATP. That generates 34 ATP, or 32 in most eukaryote cells. So that the total ATP we get from aerobic respiration is 32 to 34 in eukaryotes, 34 in prokaryotes, trying to get my mouse to show up, 2 in the Krebs cycle, and 2 in glycolysis, for a total of 38 ATP, or in most eukaryotes, 36 ATP. You do need to know the numbers, need to know for ATP for each molecule of glucose. You should also know that you generate 2 pyruvic acids, which are then burned to CO2 in the preparatory step in the Krebs cycle. All right. Any questions about aerobic respiration? In aerobic respiration, oxygen is the final electron acceptor, taking the electrons from glucose, and then oxygen is converted into water. And that's why you need to breathe, because you need oxygen for aerobic respiration. None of that oxygen, come on mouse, is converted into CO2. All of that oxygen is converted into water. The CO2 you breathe out comes from the burning of glucose. So anaerobic respiration is similar, meaning it usually has glycolysis, the preparatory step in the Krebs cycle, and it has an electron transport chain. However, this electron transport chain is a little different, and it's a little shorter. The main difference is it doesn't have oxygen as the final electron acceptor. It's some other molecule, such as nitrate, sulfate, or an iron compound. But it depends on which form of anaerobic respiration it is for what the final electron acceptor will be. It is shorter. Like I said, I seem to recall talking about this. It is shorter, so it yields less ATP. But the yield is always somewhere between 2 ATP and 36 ATP. And this means that anaerobic respiration, generates fewer ATP than aerobic respiration, and therefore anaerobes tend to grow more slowly than aerobes. All right, any questions about anaerobic respiration? If not, we're going to move on to fermentation. Fermentation is the metabolism of glucose. But we don't generate CO2, we make a fermentation end product. And there are different end products depending on which fermentation reaction you're going to go into. Let me check something here. Am I off? We already started chapter 6. That's why this is familiar to me. We finished chapter 5 last time, and we should be on chapter 6, not chapter 5. Any questions about this? Because we've already covered this. And that's why I couldn't find my marker. It's not in that thing. Let's see if I've got a 2 here. Yeah, we were talking about culture media. Now I remember last time I talked about the effects of oxygen on microbial growth. And if you grew bacteria in a thioglycolate tube, you'd see different growth patterns in the thioglycolate tube, depending on the oxygen requirements of the organism. Is this familiar to you?
Yes.
Thank you. So we're talking about culture media, and we ended up with this slide here saying that microbes require different, have different requirements, like an energy source, a carbon source, etc. For media that we have in the lab, we have agar, which is a complex polysaccharide derived from seaweed, and it's used as a solidifying agent to make the liquid broth a solid media. Any questions about any of that? So in a broth, we usually have it in a tube. And then a solid media can be in a tube, in which case we have a slant. Let me see if I can draw this. Trying to close that down. I don't want to close that down. Probably don't want to close that down. Oh, there we go. So for a broth, we just grow it mostly in a tube. For a solid media, we can grow it in a tube, and we got 2 types. We have a deep, where the media is solid, but it just goes across straight. And then we have a slant, where the media will be slanted. And then this will be agar in here. Okay. Any question about that? I know I haven't drawn this perfectly, but it's good enough. Take that out a little bit. And then you can grow your bacteria on the slant, you would grow it here. So you'd streak this area here. And then for a deep, if you grew it on the top, you'd grow it right here. But oftentimes with the deep, you actually stab the bacteria into the deep, and then you're growing it on the stab. Petri dishes are usually solid media. Any questions about that? Agar is important because it's generally not metabolized by microbes. Initially, when Dr. Koch was using solid media, he had Jello. And the problem with Jello was some of the bacteria digested the Jello, and then you had a liquid media instead of a solid media, and the colonies would all merge together, making what he called a mess. And it was actually the wife of one of his assistants who suggested he use agar. And he tried it, and it worked very well. And we still use agar today. Agar, if you don't know, is a solidifying agent that's used in cooking and baking. And obviously in microbiology, it's used to solidify a broth. Agar will solidify when you're cooling it around 40 degrees Celsius. You don't need to know this, but to liquefy it, you have to bring it up to a boiling temperature, 100 degrees Celsius. There are different types of culture media, and I mentioned that if you were in the lab, I'd show you a book, and we have a book about this thick, with each page being a different media that you can use in microbiology. There are chemically defined media where the exact chemical composition is known because we've added all of the ingredients for the media. Like this is a fairly simple media. It has glucose, it's both the carbon source and the energy source, ammonium phosphate, sodium chloride, magnesium sulfate, potassium phosphate, and then water, and that would be distilled water. And then we know that these are the only molecules in the media. This is a chemically defined media, and many organisms cannot grow in this media. If they are fastidious, they would not grow at all because you have to add certain ingredients to it. This is more what fastidious media would look like. That's chemically defined. Glucose, starch, sodium acetate, sodium citrate, oxaloacetate, and then a whole bunch of salts. I'm not even going to go through it, but that provides potassium and phosphate, potassium and sulfate. There's more sulfate and magnesium, ammonia, calcium, iron. And those are just providing minerals. But then we have cysteine, arginine, glutamic acid, asparagine, cysteine to provide certain amino acids and other growth factors shown here. Okay. And this would be something you'd have to grow for make for a fastidious media if you wanted it chemically defined. Where you do know all the molecules that you're adding, of course, you're adding also sodium thioglycolate and water, but you know all the additives to this media. So it's chemically defined. It's normally, a chemically defined media is normally only used when we're growing an autotroph, because an autotroph can make the molecules that the organism needs. So it can make the molecules that it's on its own, provided you provide everything that it needs. Like cyanobacteria could grow in this media. It may not even need glucose because it can make its own glucose. You would need to provide ammonia, phosphate, magnesium, and sulfate. Maybe potassium too, I don't know about that one. But you'd have to provide those for it. And then light. And then it could make all the molecules it needs from water and CO2, and then whatever you're adding in the media here. But most organisms require more than that. There are a few that might grow in this media, like I think E. coli would grow if you gave it glucose as its carbon source and its energy source. One that I know for a fact is one that we're using in the lab. What the heck is the name of that now? Aerogenes. Drawing a blank on his genus name though, but it's Aerogenes, and it can grow in glucose. So there are some that can grow in there, but then there would be many that would not be able to grow if glucose was the only carbon source and the only oxygen source. Excuse me, only carbon source and only energy source. Trying to think of one that would not grow. I know most Streptococcus species would not grow if all you did was supply this media. Most of the organisms that we have in the lab grow in a complex media, which we do not know what all the molecules are that we add to it. A complex media contains extracts and digests of yeast, meat, and or plants, and those provide the vitamins and organic growth factors that the organisms need, as well as a chemical carbon source and not electron, but energy source. And this would be one complex media where we're adding peptone, which is a partial protein digest. You don't need to know that. But the peptone is providing the energy, carbon, and because it's got protein, it's got nitrogen and sulfur in it as well. So the peptone is providing all of that. The beef extract is providing all of that also. And then we have sodium chloride to give some ions and to make the tonicity correct. And then agar to make it a solid media. If we had a broth, we wouldn't have agar in there. The point is we don't know what all of the molecules are we're adding because the beef will have whatever molecules the cow had, and then the cow will have whatever molecules that the cow has been eating. And so we do not know all the molecules that we're providing in a complex media. And then different complex media, especially on different lot numbers, will have different molecules because some cows will be eating, I don't know, sage or sageweed, and then other cows will be eating grass. We use complex media usually to grow heterotrophs of even bacteria because they generally require an organic carbon source like peptone or the beef extract. Any question about any of that? So if you recall, I briefly mentioned a general purpose media. All general purpose media are complex media. And this one right here is one of the common general purpose media. This is tryptic soy agar. We have different types of media besides complex media, which is a general purpose media, and then a chemically defined media. We have different types. There's anaerobic media, and this media is important for growing obligate anaerobes, organisms that must avoid exposure to oxygen. In the media, it has a reducing agent, such as sodium thioglycolate, which is a chemical that combines with and removes oxygen from the media. And the result is an oxygen depletion, at least in the bottom of the tube. You heat the media not only to sterilize it, but when you're heating the media, you drive off dissolved oxygen in the media. And then the thioglycolate removes the remainder of the oxygen. Now, once you take it out of the autoclave, oxygen will move into the thioglycolate tube, but only the top will be fully oxygenated. As you go down the tube, you'll have less and less oxygen. And at the bottom of the tube, you'll have essentially no oxygen. Another type of media is a selective media, and it has chemicals in it that halt or inhibit the growth of a particular group of microorganisms while not affecting the growth of another group of microorganisms. One example of a selective media is media that we call 7% sodium chloride media. And all it is a regular general purpose media where we've added 7% sodium chloride. Most organisms, or I should say most bacteria, cannot grow on 7% sodium chloride. And so this will select against them. But other organisms that can grow in 7% sodium chloride will grow, and we say they are selected for. An example of that would be a staphylococcal species. Can survive at 7% sodium chloride. Did I mention that most organisms cannot grow above about 2% sodium chloride? And that would be something like Streptococcus. Any questions about any of that? All right. Another media we have is a differential media, and it provides differences that you can observe between two different species. The differences are not related to how well the organism can grow on the medium, but the media has something in it or does something to make two different appearances, making it easy to distinguish colonies of different types of microorganisms. There might be a color difference between the cells, or there could be something happening on the plate, which is what happens with blood agar. Blood agar is a differential media, and it allows us to differentiate between different forms of hemolytic activity. Hemolysis is the lysing of red blood cells. With blood agar, we can streak the bacteria on the plate and see three different hemolytic patterns. There's gamma hemolysis, and here we have the white streak of the bacteria, and that's a Greek gamma. And we can see that the red of the red blood cells go all the way up to where the bacteria are. And so this is gamma hemolysis, which is no hemolysis. Here, also in white or off-white, is where we have the bacteria. And when we look at the red blood cells, they're red up until right before where the bacteria is. And we see this dark ring surrounding the bacteria. And you see it very noticeably inside the alpha that it's dark. And this is alpha hemolytic activity or alpha hemolysis. And it's also called partial hemolysis, where some of the red blood cells are lysed. That makes the blood look darker or take on a greenish appearance. And this one isn't looking very green, but it does look darker. Any questions about any of that? All right. Oops, I'm moving the picture. I didn't mean to do that. What I meant to do is go like that. The third hemolytic activity is beta hemolysis. And on the beta in white, we have the bacteria. Beta hemolysis is the complete lysis of the red blood cells. And here you see this clear region around the beta and inside the beta. That is from beta hemolysis, the complete hemolysis of the red blood cells. If you were to look through the plate right here, you'd notice you could actually view through this region here and this region here. You won't be able to view through the plate here or wherever the bacteria is, but right there you could view through the plate. And what I do is I often will hold it up to the light, and if I see the light bulb or the clock, then I know that that's beta hemolysis. Any question about blood agar? All right, let me state that normally with beta hemolysis, you have a ring of alpha hemolysis surrounding the beta hemolysis before you get no hemolytic activity where the red blood cell is normal. And this is normal, where we have a darkening of the blood, which is from partial hemolysis, which is a ring around the beta hemolysis. Any question about any of that? All right, another plate type of media we have is selective and differential media. These plates are both selective and differential. An example of that would be an eosine methylene blue plate, or EMB. This plate is selective against gram-positive bacteria. The reason why is actually because it has eosine and methylene blue in it, and this gets in the cell walls of gram-positive bacteria and then prevents them from growing. It's differential on the basis of its lactose fermentation, meaning lactose is added to the EMB plates. And if the bacteria can ferment lactose, and if it can grow, I guess I have to state that, then you will see that the colonies will turn black or they will turn greenish on EMB. And that's because of the fermentation of lactose. I'm trying to remember, I think if there's a lot of lactose around, it's greenish. And if it's black or dark, there's some lactose around. And what we're having here is a little less lactose where we have individual colonies than when we have a streak. If there's no lactose fermentation, the colonies will be their normal color, which here you can see they're white. Any question about any of that? So yes, the plate or the media can be more than one type. EMB plates are both selective and differential. Blood agar is also two types. It is differential as shown here, but it is also an enriched media. And that allows us to grow fastidious organisms, organisms that would not grow on a general purpose media. And that could be like Streptococcus pyogenes, which would not grow in a general purpose media, but it can grow on blood agar. There's one other type of media that we call an enrichment media. It encourages the growth of a certain group of bacteria. So therefore it is selective, meaning it's selecting against other cells and encouraging the growth of certain cells. And although it's selective, we call it an enrichment media because we're using this specifically to enrich for certain microbes. Such as when you're looking for a desired microbe and its presence in your sample is very rare. You have to increase the number of that rare sample to something that's high enough that you could then streak it out and get the organism. So it increases the numbers of desired organisms to detectable levels. It's often used when we're working with soil microorganisms or other environmental microorganisms or a fecal sample where we're looking for a specific organism that's at low numbers, and there's many other organisms, microbes in the soil or in the fecal sample. And this you might run into if you take a fecal sample. Obviously, there's lots of bacteria in your feces. And if we're looking for a human pathogen, it's probably not going to be very numerous. And so we put it in an enrichment media, and then we have a chance of finding it. An example of an enrichment media is when we're looking for a soil sample that degrades phenol. There would only be a few phenol degrading bacteria and hundreds of thousands of other bacteria in the soil. But what you can do is put them in an enrichment media that selects for organisms that can degrade phenol. And one way to do that is simply make phenol the only carbon source and the only energy source in your enrichment media. And that will enrich for phenol degrading bacteria. And then you transfer the sample into that flask, and then to enrich further, you take from that flask and put it into another enrichment media flask, which would be the same. And let me draw that out. Kind of, we can use this. Just erase that. So we're going to make this a liquid media now. Let me erase that too. So I need to put this across. Oops, that's not very good. Straight line, but I never claimed to be an artist. All right, so if we have an organism, which is, let's say, its presence is only one cell and 1 times 10 to the 5th cells. I don't know if I can, get that right. I don't know if I can do a superscript here, but we'll try. Come on. For some reason my paint isn't working, but here it's going now. Well, the font isn't what I'm looking for. So let's just make that smaller. And let's see if I can blow that up. Well, there. This sort of will do it. Yeah, about. So we have 1 cell and 1 times 10 to the 5th. We can put it in the enrichment media, and let's say it increases it tenfold. That's good. So now we've increased it to 1 times 10 to the 4th. I think I'm going to go just 10 E4. We can then take from this media and that back, take a sample out and put it in the same media, but another tube of it, and once again we enrich it, and now it's going to be 1 cell and 10. Oh, I don't have the one cell over, I'm just going to go 1,000, 10 to the 3, okay? And after that, oh, wrong one. Bring this over. We can transfer from this tube and put it into that tube, and then increase it to 1 in 100. Now, because we have 1 in 100, it would be possible taking this out and then streaking it on a petri dish. And then we have a chance of getting a colony, which would be the bacteria of interest. This is still not good because you have to streak out 100 colonies just to get one. But if we did it one more time, assuming that we have a tenfold increase each time, then we'd have 1 in 10, which it would be very easy to streak it out. You'd only need 10 colonies, and then one of them would be of interest to you. Any question about how you do an enrichment media? All right, so this slide is showing you the different types of culture media: chemically defined media, the complex media. Most of the media that we use in microbiology are complex media. Okay? In fact, now that I recall, I think I've only used one, maybe 2 chemically defined media. Glucose salt media was chemically defined. All the other were complex media, a reducing media for growing anaerobes, selective media, differential media, and enrichment media. And please understand that you can have combinations, like blood agar is a complex media that is also differential, and you could say it's an enriched media, meaning that it allows you to grow fastidious organisms. And don't confuse enrichment from enriched media. Blood agar is an enriched media. It is not an enrichment media. An enrichment media is a selective media because it does select. And what else? There's also differential media, and you can have combinations like selective and differential. All right, so the goal of streaking for colony isolation is to get an isolated colony. Now, once you have an isolated colony, you can then take that colony and streak it out for colony isolation a second time. And then when you get a isolated colony that's gone through 2 colony isolation events, you have a very good chance that you'll have a pure culture, a culture that contains only one species. If you were only to streak it out one time, you would not have a great chance of getting a pure culture. It would only be about 70% of the colonies would be pure of only one species from a mixed culture. And if you do it 2 times, the chance that it is a pure culture is greater than 95%. Any questions about any of that? The colony, of course, is a population of cells arising from either a single cell or a group of cells that are stuck together. Because we don't know if the colony was formed by a single cell or a group of cells, instead of saying the number of colony forming units, I mean the number of cells that got started in the original solution, we say the number of colony forming units there are in the original solution. So if you have a plate that you dilute the sample out and you get only 30 colonies from it, it's not quite correct to say that that sample had 30 cells per mil, because some of the colonies may have been formed by more than one cell. It is more correct to say that the number of colony forming units per mil are 30. Did everyone get that? All right. So obtaining single colonies is a critical part of obtaining a pure culture. And how we do that is using the streak plate method, AKA streaking for colony isolation. And to get the pure colony, you need to streak it out 2 times. Any question about any of that? So here is the streak plate method where we set up, let's see if I can blow this up, where we set up the plate and then we streak it out in 3 sectors. Initially, we grab from our stock culture and put the bacteria on a sterile loop, streaking it on the plate here. And then we sterilize the loop, killing the cells that are on it. And then we streak out sector 2 here. And the goal is with sector 2 to overlap a little bit of sector 1. And this way we pick up cells from sector 1, which are then streaked out in sector 2. And then you do the same for sector 3, which in reality it should be coming up this way. And then this would be the 3rd time. And then streak out sector 3. This way for sector 3, it will be much more dilute than sector 2, which will be much more dilute than sector 1. Now, in each of the sectors, you are doing some dilution, and that is the cells will be most concentrated in this region of sector 1, and they will be diluted in this region of sector 1. And so when you do the colony streaking method, you want to make sure that when you're setting up sector 2, you don't go way into sector 1, getting the most concentrated cells. You want to get the lesser concentrated cells from sector 1 and then carry those to sector 2. And the same thing with sector 3. You don't want to streak way down here, which would be, well, actually right here, we don't have anything. So that should be a little bit, this should be starting over here. But you want to streak in sector 3 where it's diluted on sector 2. You don't want to go way back into sector 2. You want to do where it's already diluted in sector 2 to pick up cells and then streak out in sector 3. The reason for doing this when you're doing colony streaking is you're getting dilute bacteria. It's diluted from here to here, and then it's diluted in sector 2 further from sector 1, and then sector 2 is diluted in this region from this region, and then in sector 3 it's diluted from sector 2, and it's diluted also from this region to that region. And that's probably most visible. See if I can blow this up a little. Most visible with sector 2. It's more concentrated here than it is here. And then this is sector 3, which is more concentrated here than it is over here. The goal of the colony streaking is someplace along the line, get the bacteria diluted enough that you are streaking out individual cells where they will grow up into individual colonies. You do not have to get the individual isolated colonies in the last sector. If it happened in sector 2 or even in sector 1, it's fine. The point is somewhere along the line, you get isolated colonies. Any question about any of that? And actually, let's blow this up again. This is a little better shown than here, and that is it's most concentrated here. And to set up sector 2, we're not going way back into there. We're getting it where it's more dilute. Any questions about any of that? All right, when we're talking about the growth of bacteria, remember that we're referring to the increase in number of cells, not the cell size. There are 3 ways that bacteria can grow. Most bacteria grow by binary fission, where one cell just splits down the middle into 2 cells. Binary fission. But there are a few bacteria species that grow by budding. And that's where the parent cell puts out a bud. Let's see if I can draw this. So we have the parent cell here. And what it does is it grows a bud right here. And with time, I'm going to draw the next one down here, that bud grows. And by the way, initially that bud is not separated from the parent cell. So let me put a hole there. And this bud grows. Try and make it bigger. And with time, it just continues growing. And that'll happen until the bud becomes similar in size as the parent cell, in which case it usually will separate from the parent cell, meaning cell membrane and cell wall will start growing there. and then it'll separate. So that's budding, where cells form from a small outgrowth called the bud, and then enlarges with time. It separates when the bud becomes about the same size as the parent cell. There is a third way that bacteria can grow, and that is by reproductive spores. Let me get a new page here. And here we have the parent cell, and it usually will be nice and big and will be standing up. We'll put it like that. And it'll grow buds, usually at the top. And usually there's 4 of them. I don't think I'm going to be able to get 4 here. Maybe I got 4 here. It doesn't matter how many there are, but there's buds. The buds are smaller than the parent cell. And the point of the bud is for it to be broken off or carried by animals or something like the wind. And then the bud can go elsewhere and find a nice environment and over time grow, in which case the bud will start growing into its own parent cell, which we would call a vegetative cell. This is called a reproductive spore, not to be confused with a endospore. Reproductive spores are usually at the tip of a filament, so that the reproductive spore is more likely to scatter. And it is reproduction because one cell makes more than one spore, so that we can get a potential later on of 5 different cells from this one cell. And this one cell is 1, and then these spores grow into 4 other cells, vegetative cells. So that is reproduction. If you recall, an endospore, you have only one endospore from one parent cell. And then the parent cell dies, the endospore continues. So it's 1 to 1. That is not reproduction. So don't confuse a reproductive spore from an endospore. And generally speaking, I will always use the term endospore or reproductive spore. Any question about any of that? All right, let's talk a little bit more about binary fission, the way most bacteria grow. What happens in binary fission is you get a parent cell and it replicates its DNA. So there's 2 copies of the DNA molecule. The DNA, one moves to one side of the cell, the other moves to the other side of the cell. The cell then literally starts growing a cell wall and a plasma membrane between the 2, between the 2 DNA molecules, literally starting to split the cell in 2. And this is the artist's rendition. This is an actual electron microscopic image of a cell going through binary fission. And here we see the cell wall partially forming. And there's the DNA molecule on one side of the cell, the other DNA molecule on the other side of the cell. The cell wall grows and literally grows between the 2, creating 2 daughter cells from one parent cell. And then the cell will then separate. Any question about any of that? Let's look at budding. We're going to look at it in a yeast, but it happens the same in bacteria. But we'll look at this cell here, and there you can see it's starting to form a bud right there. That bud enlarges, the DNA molecule divides, and right there you can see the DNA moving into the bud, and the bud's getting larger, larger, and larger, and then about right there, that size, it starts separating. And then both this cell and that cell can start forming a bud. This one is growing here. This one is starting to grow there. All right, any questions on budding? When we're talking about bacterial growth, it's important to discuss the generation time. Students often have trouble with the generation time, even though they're familiar with the generation time with the human population. Generation time is the time required for a single cell to divide into two, or it's the same as the population to double in size. So if you have one bacteria cell and the generation time is 3 hours, that cell in 3 hours could divide into two. But if you had a population of cells, let's say 1 million, the generation time to double in size would be 3 hours to generate from 1 million to 2 million. Anyone have a concern about that or question about that? The generation time varies considerably between species. It can be as fast as 20 minutes per generation. Like E. coli had a generation time of 20 minutes. But most bacteria are a little slower than that. Most have a generation time between 1 hour and 3 hours. There are some which take much longer, like Mycobacterium leprae, takes 10 days to double in number. So its generation time is 10 days. One of the reasons why Mycobacterium leprae grows so slowly is it has mycolic acid in the cell wall. Mycolic acid is a very complex lipid, and it takes the cell time to grow its cell wall. What students get wrong at the generation time is its units. It's a time per generation. Like E. coli, we say its generation time is 20 minutes, but when we say that, it's implied that we're talking about 20 minutes per generation. So for most bacteria, it is 1 to 3 hours, but it's 1 to 3 hours per generation. Okay? And students get that wrong, maybe because they're so used to miles per hour. So they're used to talking about traveling at a certain speed, and then you go a certain number of miles per hour. Well, this isn't generations per hour. It is the number per generation, meaning the time per generation. Any question about any of that? So if I ask you what is the generation time, you know to state it is the time per generation, not the generations per hour. Any question about generation time? If not, let's graph the generation time. Microbiologists like to use a semi-log plot when they are graphing the generation time. If we did a regular arithmetic scale where we'd have the numbers of cells on the y-axis and the number of generations on the x-axis, if we used a regular arithmetic scale, we'd have this plot shown here, which all the points really, most of the points will be right here, all clumped together. All the points, not all the points, but a lot of the points. Like this would have 1, 2, 4, 8, 16 before you get 32. And then the graph starts rising very quickly, meaning it's an exponential graph. And the next point in the generation time will be, I guess that's the last point. The next point will be on the ceiling here because the graph is exponential. If on the other hand, we were to use a semi-log plot where we put the log number of cells on the y-axis and then the generations on the x-axis. So the generations is still arithmetic scale. It's only the number of cells, which is a log scale, why we call it semi-log. If we were to do that, we would separate out these points here much better. They're not shown here, but it would be something like 1, 2, 4, 8, 16. And then that's probably presumably close to 32. So they're not all bunched together. They are separated out. And then our next point here will actually be about right here. So it's not on the ceiling. It's actually close to being on the graph. You could extend the graph to get the next point. So it allows us to separate out the points in this region. And then it allows us to get more points on the graph. And if we extend this graph, we can easily get the next point on. But the arithmetic scale, we would not be able to get the next point on because the next point is probably on the ceiling of my apartment anyways. And then the second reason why microbiologists like doing the semi-log plot over the arithmetic plot is when we describe this mathematical equation for the growth, it's very simple. It's a y equals mx plus b graph. So it's a very simple to get the equation and then try and figure out what, for this time here, how many cells there are. You can mathematically do it if you know the equation here. And this would be 20 generations, 6 log numbers over 20 generations. So that's the y equals mx part. The b is the intercept point, which in this case would be very easy because it's at 0. On the other hand, if we were to do it this way, we'd have a very complicated mathematical formula for describing the growth of bacteria cells. Let me see if I can remember how to do this. Y will equal mx plus nx squared plus b for this mathematical equation. And that would be difficult trying to figure out where this point goes at this time, just because it's not a linear relationship. And with calculators, you could do it, or with a computer. But like I said, it's much easier doing it on the semi-log plot. Any question about any of that? All right. When we're talking about the growth of bacteria, it's important to realize that bacteria have 4 phases of growth. And this is generally true of all populations of living organisms. Generally true. Like they introduced deer to an island once. And initially, the deer went through a lag phase and then had an exponential growth phase. And then they started eating all the food that was available. So they entered the stationary phase. And then when that food was gone, they entered the death or decline phase. Okay. But for bacteria, we tend to find this type of a bacterial growth curve. Like when we're growing bacteria on solid media, and then we grow it on a liquid media, we will get this curve. What happens in the log phase is right here, we're inoculating the media with the cells. But the cells do not immediately grow in this new media. They enter a lag phase. In the lag phase, what the cells are doing is getting all the enzymes and requirements that they need to be able to grow in the new media. Once they have everything they need to grow, they will start growing. And then they'll grow exponentially because initially we have lots of nutrients, little bacteria numbers, and little, if any, waste. So the bacteria start growing exponentially. And there's lots of space at this number time too, because there's few bacteria. But with time, the bacteria will start slowing their growth. What's happening when the growth starts to slow is we have now lots of bacteria. There's some accumulation of waste occurring, and there's less space for the bacteria to grow and less nutrients around for the bacteria. So they start growing at a slower rate. Eventually, the population will reach a stationary phase where the population numbers will not change. Stationary. What's happening is the lucky cells have enough nutrients around them and enough space around them to grow, and they don't have that many wastes around them. The unlucky cells have lots of waste around them and few nutrients or space around them. And so they are not growing, and they are actually dying. In the stationary phase, you have the number of new cells equal the number of cells that are dying. Or you can say birth equals death. The population is stationary. Any question about any of that? Well, as you continue growing the cells, there will be fewer and fewer nutrients, more and more waste, and probably less and less space. That will bring the curve into the death or decline phase, where early on in the death or decline phase, you might have some lucky cells that are still growing, but you have more cells that are dying than cells that are growing. And so the population numbers start declining. Now later in the death or decline phase, you have no cells that are growing. The cells are either in stasis or they are dying. How long it takes before all of them die will depend on the bacterial characteristics. For something like E. coli, which is fairly easy to kill, this death will happen in a week or less, depending on where we are in the curve. But from here to there, in a week or less, you'll get all the cells dying. But for something that makes endospores, before the last endospore dies, this could be years, decades, centuries, or even millennia. But it takes a long time for endospores to die. Any questions about the phases of growth? All right. When we're talking about the measurement of bacteria, there are different methods used to measure the growth of microbial populations. There are direct methods and there are indirect methods for measuring bacterial growth. The direct methods include the plate counts, filtration, most probable number, and the direct microscopic counts. Indirect methods involve turbidity, some metabolic activity, and the dry weight. I have actually used all of these methods for getting an estimate or the number of microbes I have growing. I've used all of them except for the most probable number. Any question about any of that? All right, the first I want to talk about is the plate counts. It's one of the most common methods for measuring the number of bacteria in a microbiology lab. It is also called the viable plate count because this method of getting an estimate of the cells is the only method that counts viable cells. Everybody get that? The plate count is the only method we use to get viable number of cells, meaning it only counting the viable cells. It's not counting the dead cells. What you do is you set up different dilutions of your bacteria, and then you plate each dilution. And then each dilution plate, you incubate it for about 24 hours. And then you get growth on the plates, which will be colonies. And then we count the number of colony forming units on the plate to get the number of colony forming units per mil of our original sample. So you make several serial dilutions, and usually what you do is you take one tube, take 1 mil of your sample and put it into 9 mils of media, mix it, and then you got a 1 to 10 dilution. Let me go ahead and go down here. And then you take 1 mil out of this 1 to 10 dilution and put 1 mil into 9 mils. So we have another 1 to 10 dilution. And this is together, this one here, a 1 to 100 dilution, because it's 110 times 110, which is 1 over 100. You then mix that and then take 1 mil out of that to set up your 1 to 1,000 dilution. And you take that 1 mil and put it into 9 mils of media, mix it, take 1 mil of that and put it into 9 mils to get your 1 in 10,000 dilution. And then if you can do it again to get your 1 in 100,000 dilution. Then you have to plate each dilution onto a Petri dish. There are different ways of inoculating the Petri dish. You can do the pour plate, pouring on 1 mil and then spreading it. Or you could use the spread method where you put the media on and then you spread it around with the spreader. You then incubate these plates and allow the colonies to form, and then take a look at them. You'll notice that some of the dilutions, the 1 in 10, the 1 in 100, the 1 in 1,000, have too many colonies to count. Even this one, it'd be very difficult counting the number of colonies right there, for example. And these two plates, we have countable colonies, but one of them is better to use than the other. And I'll explain that in a moment. And then you do your counts to obtain your original, the inoculum, what the concentration of the cells were per the original inoculum. And this is about 30 to 40 cells. The dilution is 1 in 10,000. So you multiply by the inverse of this, 10,000 over 1. And that would be 30 to 40 times 10,000 cells, colony forming units in your original inoculum. Any question about any of that? Go backwards. So when you're doing the count, you want to do it on this plate, not on this plate. And the reason is, let me see if I can get that to show. If you have a plate where you only have 3 colony forming units on the plate, and then you go, I probably can't do this, plus or minus. I don't think I can do that, but we'll look. Oh, yes, I can. Plus or minus 1. Oh, gotta remove the underline there. That's a very large plus or minus. On the other hand, on the plate where we have 30 colonies, and we go plus, oh, let's just grab it here. Go plus or minus 1. Well, here you're going plus or minus . Oh, that's not what I wanted. There we go. Plus or minus 1. That's plus or minus 33% for your mean. And here, I might get this wrong. That plus or minus isn't 33%. It's going to be, what is that? It's 1 over 30. 1 over 10 would be 1/10. 1 over 30 would be around 0.3. So it's something like plus or minus, oh, I don't want that. So it's plus or minus something like 3.3%. So that this mean is much more reliable than this mean. Everyone see that? So when it comes to which plate you should count a plate looking like this, where the number of colonies is greater than 20, but less than 300 colonies per plate. And ideally, you want it between 30 and 300, or 30 and 250, sometimes they say. You don't want to do it on this one because the mean will have a very large variation, whereas the mean here has a much smaller variation. Any question about how you do the viable plate count? Now, one disadvantage of the viable plate count is it does take time to get your count because you have to plate out these serial dilutions, which that's not so bad, but then you got to allow the culture to grow, and that takes at least a day. This is fine whenever the bacteria numbers in the original inoculum is very high. What do you do if the bacteria numbers is very low? You can't use the plate count method because even if we were to plate the original, you would get very few colonies, if any. Like if the bacteria concentration were like 1 colony forming unit per liter, and you were only to plate 1 mil, you'd likely get no colonies on this plate. So the viable plate count method would not work if your starting bacteria are very dilute. What we do if the bacteria are very dilute is we use the filtration method. It is only used when the bacteria are very dilute. You take a liquid media and then you put it through a filter, shown here. The filter has pores in it to let the liquid media go through, but the cells will be blocked. And then you take the filter and sterilely put it over an agar plate and then incubate it. And wherever there's a colony forming unit, you'll get a colony. And then you count the number of colonies very similar as you do for the viable plate count. All right, the filtration method can only be used when the bacteria number are very low in question. And you have to have a liquid so you can pass it through the filter. There's also direct microscopic counts where somebody counts the cells under a microscope. You use a special gridded slide that allows a specific volume of liquid underneath it. So you have a slide where there's a measured volume of liquid. And then you count the cells under the microscope. As shown here, there's the gridded slide, and it has a specific volume under it. You add your inoculum right here, and it'll be whipped by the cover slip. And then right here, you count the number of cells and this square right there. Usually the slides are designed so that the number of cells you have in this block here will then be 1/1,000,000th what you'd have per mil of your original sample, meaning it's 1 microliter like liquid that is the volume that you put under here. And anyways, you multiply this number times 10 to the 6. That's usually the way the gridded slides are set up. Here we're looking at the slide, the gridded slide from the side, and you can see that it has a cover slip on it that makes a specific volume right there. And then you add the liquid right here, and it's usually pulled or whipped underneath the cover slip. So usually you don't need to do anything to get the liquid to go under the cover slip. One problem with the direct count method is if you have live cells that are moving around, it'd be very difficult to count the number of cells because this cell could start here and then move down there. And then these two could be off the grid that you're counting and then could move into the grid. So with cells that are motile, it is very difficult to use the direct microscopic count method. There are tricks you can do, like set up your slide and then let it sit until the cells start dying. Or you could add something to thicken the media, and then the cells, even though they're swimming around, they don't go very far. And so you can count the cells that way. All right, so that's all of the direct methods I'm going to discuss. The plate count method we talked about, filtration we talked about, and then the direct microscopic count we talked about. I'm not going to talk about the most probable number. So if you want to read about that, you can read about it in your textbook. One of the reasons why I'm not going to talk about this is the way that is least used in microbiology. And it's the most complex method for getting your cell count. All right, there are indirect methods for getting your cell count, and they use indirect methods for getting an estimate of the number of cells. And there's 3 indirect methods. The first is turbidity, where you have a liquid culture and you take the media, put it in a test tube, and then shine, excuse me, there's the media there, put it in the test tube, and then shine light through, and then measure the amount of light coming through the media in the spectrophotometer. You then take your sample with bacteria and put it in the tube. Usually what they do is they just fill the tube with the media. And then you shine the light through it, and less light will get through because the bacteria will stop the light from coming through the media. And then you measure the amount of light coming through the media of the tubes with cells and compare it to the tube without cells. And then you get your estimate of the number of cells. Usually what you do is you get the absorbance reading here, and then you look on a chart that somebody's already made to get your estimate of the number of cells. And usually if you're using the turbidity meter, you're given a solution that has so many plastic microbes in it. And then you can set the chart based on that. So it already tells you how many cells there are per mil, and then you measure absorbance, and you can set up your chart for what it will be for determining how many cells there are. Anyways, this is a very quick method for getting the estimate of the cells. It's only an estimate. And what you do is you put your bacteria in a tube, and then you put your media in another tube, and then you get your absorbance estimates, and then compare those numbers to a chart, and that gets you the estimate of how many bacteria cells you have per mil, based upon how much light is absorbed. Any question about any of that? Another way you can get an estimate of the number of cells is by doing metabolic activity. Here you're assuming that a bacterial cell makes a certain amount of metabolic product per time, such as CO2. And then you measure the CO2 that is accumulated over a given length of time to get your estimate of the number of bacteria. For example, if your cells of bacteria make, I don't know, 1 microliter of CO2 in an hour, and then you run this for several hours. This might be a bad example because it's going to be so long. But let's say you run it for a day, and then you measure the amount of CO2 that you have, which in this case would be, is it 24 microliters CO2, something like that, and look on a chart, and then get an estimate of the number of cells that made that CO2. So the more CO2 that's made, the higher the concentration of cells you have. And like I said, you have to have a chart already set up, meaning somebody's had to measure the number of cells, probably by a direct count, and then measure the CO2 produced. And then you know what your counts are from the metabolic activity. Any question about metabolic activity? All right. The third way of getting an estimate of the number of cells is by dry weight. You take your cells, what you've been growing, and you centrifuge it. And the cells will centrifuge to the bottom of the tube, and then the liquid media will be on the top. You just pour out the liquid media, and then you dry the cells. Now that takes a little bit of time to dry the cells, but you then can, whenever the cells are dry, measure the weight of the dried cells. And then the more cells you have, the more dry weight you'll have. And you look on a chart and you determine if the dry weight is, I don't know, let's say it's 2 grams, how many cells you have in your original media. One problem with all of the, well, the dry weight anyways, is that you're going to measure the dead cells as long as the viable cells. And that's true for the spectrophotometer too. You can't tell, is this cell here viable or is it dead? And like I said, the best way for determining and counting the viable cells is to do the viable plate count, because it only counts a cell if it can form a colony, and therefore the cell has to be viable. All right, any questions about estimating bacterial numbers? If not, I'm going to, oh, wait a minute, this is the 4th. Today is Thursday. Yeah, let's go ahead and start chapter 8. We've got a little bit of time here. So chapter 8 is genetics. The major goals and the rough lesson outline are shown here. Know the terms. What's a genome? What's a gene? What's a genotype? What's a phenotype? What is vertical and horizontal gene transfer? What's an operon? What's a promoter? What's an operator? What is a structural gene? What is crossing over? What is transformation, conjugation, and transduction? Be able to describe what DNA replication is, RNA transcription, and protein translation. And then lastly, understand the different types of mutation. Any questions about what we're going to be covering? All right, this is a summary of what we're going to be doing. First, we're going to talk about the structure and function of the genetic material. We'll talk about how the genetic information flows. We'll then talk about DNA replication, DNA transcription, and DNA translation. And then we'll move on to talking about the regulation of bacterial gene expression, talking about what an operon is, how operons are controlled. We'll then move on to talking about what a mutation is and how you get mutations, talk about genetic transfer and recombination. And then lastly, we'll end with genes and evolution. All right, genetics is the study of what genes are, how genes carry information, how that information is expressed, and how genes are replicated. A gene is a segment of DNA that encodes a functional product. That functional product is usually a protein, but it can also be an RNA. like the tRNA gene, only codes for tRNA. There is no protein that's made. Let's see if I can blow this up. So the DNA is on the chromosome, and there will be a certain region of the DNA which codes for a gene, and this codes for a functional product. And there we're seeing a map of the human gene. This is actually the X chromosome and some of the genes on the X chromosome. The genome is all of the genetic material in a cell or an organism, if we're talking about something like the human genome. But for microbes, generally the genome is the amount in the cell because that's all they have. They don't have an organism made of several cells. The study of genomes is called genomics. And when we're studying the genome, there are two things you need to know. You have the genotype, which is the actual genes of an organism for a trait. Like for this individual, they have blue eyes because their genotype is lowercase b, lowercase b. But the individuals who have brown eye have the genotype either capital B, lowercase b, or they can be capital B, capital B. And the lowercase b is recessive, why it is lowercase. The capital B, which is brown, is dominant. And so an individual who has the genotype capital B, lowercase b, will have brown eyes. The phenotype is the expression of the genes. So for this individual, the phenotype of the capital B, lowercase b is brown eyes. And that would be the same as the individual who had capital B for their genotype. Their phenotype will be brown eyes. The only individuals who have blue eyes are those individuals who have genotype lowercase b, lowercase b. Any question about that? So the genotype does determine the phenotype, but the phenotype will be the expression of the gene. And I guess when you're talking about capital B, lowercase b, the lowercase b, that gene is either not expressed or its expression is hidden. So we don't see the blue in there. Here we're seeing the chromosome of E. coli, and it only has one chromosome. And obviously it's pretty big. All right, any questions about any of that? If not, I think we're going to stop here and have the lab at 7. If there's no questions, I'm going to say goodbye. And
I have a
see you-
question, but it's about an email I sent you. I can wait until everybody leaves.
Okay. All right, I can't close this down with you on here.
I can also ask you after lab if that's easier.
Yeah, what I'll do is I'll close this down. No, I can't open the lab because I'll start recording. Why don't you ask during the lab, and if that's okay with you,
Okay,
that'll give me a chance to look at the email. I haven't.
Okay, thank you.
All right,
thank you.
all right.
I can ask it now if that's easier, or is it, you
Yeah,
still
you
want me
can
to wait?
ask it now. Let me stop recording.